Hello friends, today I will give you the idea about the pharmacognosy practicals. How many practicals are there, how they are going to be performed, and also we will cover the first experiment in this uh, pharmacognosy subject also. Now, about the idea about how many practicals are there, we will see. So, this again I repeat that while listening or watching the video of practicals, you have to refer the lab manual of that respective subject. So, now before listening this video of pharmacognosy practical, you open up your lab manuals of pharmacognosy subject and go through that also, and in index you will find that these pharmacognosy practicals are divided into four sections. So first part is of three experiments in which the first is about the compound microscope, second is about the technique of section cutting, isn't it, staining and mounting, and third is about the various reagents or microchemical reagents also we can call, isn't it? Then second part is of histological practicals, that is the anatomical study are is to be carried out of those drugs which are given. In this total 10 experiments are there, isn't it? Then in C part, the morphological studies. So morphological experiments are there. These are total 7, isn't it? Then part D, that is the last part in this, the identification test of crude drugs. So to identify the drug by physical and chemical test, in this 5 experiments are there. So whatever the crude drugs, what are these crude drugs, how many we are going to study, all this you are knowing in the theory. I have given you idea. So these drugs we are going to cover in practicals also, and the practicals are divided into three main sections. First is introductory section about microscope. You go through that, you are already knowing about the microscope, you have studied in your HSC, isn't it? Regarding technique, I will give you idea and the reagents that are used, isn't it, in studying the histological characters of crude drugs, isn't it? Total 22 experiments are there. So in that 10 experiments are of transverse section, isn't it? Then 7 experiments are the morphological study of crude drugs, and 5 experiments are of chemical test, isn't it? Now, regarding introductory practicals, I will give you idea that how the section cutting is done, isn't it? Because before starting with the second part, that is the histological practicals, you must know idea about how the section is taken, isn't it? Now, the section is to be taken of the organized drugs, isn't it? Organized drugs means you are knowing about organized and unorganized. So again I clear that this section is possible of organized crude drugs. That means the drugs which are representing direct part of the plant like fruit, seed, bark, leaves, fruit, isn't it? But when unorganized drug is there, when semi-solid drug is there, when any resinous substance is there, so the drug can be studied by the physical or chemical test, as I have given you in section D, that is the part D practicals, isn't it? Now this transfer section is obtained by cutting the drug. How it is to be done, that I will give you idea, isn't it, later on. By this you just try to understand that how the section is taken. So whatever the sample is there, that is to be taken in between the two fingers, and it is to be cut with the help of blade, new blade, isn't it? So the act or process of cutting or dividing parts from a fixed mass of tissue, isn't it? So simply this whatever the sample is there, that is to be taken in between two fingers, isn't it? So thumb and forefinger, in between that the drug is taken and repeated sections are taken and they are transferred in the watch glass, isn't it? You must refer your lab manual also, so that you will get idea, isn't it? So whatever the repeated sections are taken, then they are transferred, isn't it, into the watch glass containing the water, isn't it? And why water must be present in watch glass? Because if you are not transferring the sections in water, it will get dry. So whatever the section you have taken, once it becomes dry, you will not be able to study the histological characters. You will not see any cellular arrangement of that section, isn't it? Because it will get dry. Therefore, these sections are to be kept in water, isn't it? So after sectioning, how the very fine sections are there, they are to be placed on the slide, isn't it? So by naked eye, we cannot see the characters. For that, microscope is needed. So whatever this figure is showing, in first figure you will see that the slide is there, the fine section is taken on the slide, then you have to add a drop of staining solution also, isn't it? So staining solution is very important, because without staining solution you cannot use, or you cannot see the characters, isn't it? So in sectioning there are different steps that have to be followed, isn't it? So in experiment 2 you can go through that and see that what are the various steps that are to be followed and what are the requirements of the histological practicals. That is also given. You must read that so that you will get the idea, and after that only you will understand whatever I am trying to explain you, isn't it? So the sample is to be prepared for sectioning. Whatever the sample is there, that is boiled in water. So by that, whatever the tissues are there, that becomes soft, isn't it? So boiling of the sample is very necessary. Section cutting, as I gave you idea. So before section cutting, boiling of the sample is very important, isn't it? After taking section, after sectioning, the fine section is to be taken on the slide, isn't it? Selected pieces that are to be taken on the slide, add staining solution, isn't it? Staining is very important. Now what is staining solution? So staining solution is nothing but a particular stain or a dye, or which gives color, so which combines chemically or physically with the cell content, with the drug, with the section, to give its own color, isn't it? So staining process is also very important. Go through the manual, you will get the idea about the staining process, isn't it? So you have to add staining solution in the watch glass in which number of sections are there, isn't it? And then with the help of brush you can transfer the section from watch glass to the clean slide, isn't it? And on slide you can add one drop of staining solution, isn't it? And then with the help of filter paper you can remove the excess staining solution also, isn't it? Then you have to mount the slide also. Now how mounting is done? You have to transfer the section on the glass side with the help of brush. So camel hair brush is needed for mounting of the slide, isn't it? Then you can add one or two drops of water on the section with the help of dropper, isn't it? Then place a clean cover slip over the section with the help of forceps and needle, isn't it? Again with the help of filter paper or blotting paper, remove the excess stain or excess water, isn't it? And then this slide is to be observed under 10x lens, isn't it? Now, when you will observe this slide under 10x lens, so the second figure you can see that how the cellular arrangement is observed, isn't it? How the cells are arranged, isn't it? The study of those cells and various characters is carried out, isn't it? This is about the staining as I gave you the idea, isn't it? Go through the uh, third part of the experiment also in introductory, where there are various examples of microchemical reagents, isn't it? So they are useful to study the various characters or specific character of any drug can be studied, and these reagents can be used on either section or on the powder drugs also, isn't it? Now, the introduction we have already covered. Now, the first experiment under first category, that is the anatomical study of crude drug. In that, the first experiment number 4 we will see, and that is nothing but TS of Cinchona bark. So, the title of experiment is to study morphological and microscopical characteristics of Cinchona bark. So, in this first experiment, you will take transfer section of Cinchona bark. Cinchona bark is a organized drug, isn't it? You will study morphological characters also pharmacognostic study, that is the synonym, biological source, chemical constant and uses of the drug. So Cinchona bark is a organized drug, and this is the slide which is showing the appearance of the drug, isn't it? So bark portion is medicinally important, isn't it? And the section of the bark is to be taken, isn't it? So Cinchona bark, the other name synonym is Jesuit's bark, Peruvian bark. In some books, Panama bark is also the another name of Cinchona bark. Biological sources, it consists of dried bark which is obtained from the plant of the species Cinchona calicea, Cinchona officinalis, isn't it? It contains not less than 6% of total alkaloids. So earlier we have studied 24 drugs. One is pharmaceutical aid, 14 drugs. Carminatives we have studied 10 drugs. Carminatives contain volatile oil as chemical constituents, isn't it? So here Cinchona contain alkaloids and it belongs to family Rubiaceae. Now, the morphological characters of the drug, the various varieties are given, and in biological source we have seen Cinchona calicea, Cinchona officinalis, isn't it? So Cinchona succirubra, Cinchona legeriana, these are another species of the Cinchona bark. So it is red in color, isn't it? Then odor is slight and characteristic, and taste is astringent and bitter. So drugs which contain alkaloid, they are always bitter in taste, isn't it? The size and shape of the Cinchona species varies from one species to another, isn't it? They are available in the quill form, isn't it? That we have seen already in the first slide, isn't it? Now this is the figure of TS of Cinchona bark, which shows the important microscopical characters after taking transverse section. Now in introductory practical I gave you idea how sectioning is done. So section cutting, mounting and staining of the slide, isn't it? Staining of the section, and how slide is prepared, and that slide is to be observed under 10x lens under compound microscope. So under compound microscope under 10x lens you will see these characters of Cinchona bark. This is the transverse section of Cinchona bark. Now the bark drug shows following characters: cork, cortex, and secondary phloem, isn't it? So first portion is cork, isn't it? So cork portion shows several layers of elongated cells which contain reddish brown matter, isn't it? Then calcium oxalate crystals are present, isn't it? Then after cork there is phallogen and phallodome. Now these are different types of cells. Phallogen, they are 2 to 3 layers, they are thin walled cells without any cellular content. Phallodome, this is 6 to 8 layers, isn't it? Without any cellular content, isn't it? So cork layer, whatever the phallogen and phallodome cells are there, they are without any cellular content, whereas the cortex region, isn't it, that shows the cellular content also, like calcium oxalate crystals are present. These cells are little bit bigger as compared to cork cells, isn't it? They contain reddish brown matter also, isn't it? So the sclerites are absent in this case. Now what are sclerites that we will study in case of TS of cinnamon bark, isn't it? Which is from carminative category, isn't it? Then secretion canals. So whatever these two bigger cells are there, they are called cavities or secretion canals, isn't it? Then secondary phloem, this is the second part of the section where the important characters are sieve tubes, isn't it? These are the compact cells, isn't it? Having narrow companion cells, isn't it? And these are collapsed type of cells. Then phloem parenchyma, which is dark reddish brown colored, isn't it? Dark reddish brown colored phloem parenchyma is there. Then medullary rays are there. So medullary rays, they are one seriate or biseriate or triseriate, isn't it? So they are elongated, isn't it? Elongated medullary rays which contains starch grains also, isn't it? These are important microscopical characters which are observed under TS of Cinchona bark, isn't it? Then chemical constituents which are present in the drug, as I said that Cinchona bark contains alkaloids, and the main alkaloids that are present are quinine, quinidine, cinchonin, cinchonidine. The less important alkaloids are cuprine, hydroquinone, and quinic acid, isn't it? Chemically, these all are alkaloids, isn't it? Alkaloid is a separate category. As I said, in carminatives, volatile oils are present. In pharmaceutical aid, acacia tragacan, they are carbohydrate containing drugs, isn't it? Or some are gelatin containing drug like gelatin, isn't it? Protein containing. So gelatin is a protein, isn't it? So cinchona bark is alkaloid containing drug. Uses are, this drug is anti-malarial. So the drug which is used in treatment of malaria are called anti-malarial drugs. So it is used in malaria. It is used as antipyretic, that means to lower body temperature, isn't it? Used in various preparations like tinctures, extracts. Then it is a stomachic also, antipyretic, as I said, to relieve fever or body temperature. It is also useful in arrhythmias and in atrial fibrillation, isn't it? So these are various preparations that are displayed on your slide, isn't it? Of these cinchona bark. The various preparations, as I said. So cinchona is used in the form of extract also, and also it is used in the form of tinctures also, isn't it? So in this first experiment, that is experiment number 4, we have covered the morphology of the drug, microscopical characters, isn't it? As well as the cognostic study of this drug also, isn't it? So I hope you have understood the introductory part of this pharmacognosy practicals, and in this I have covered this first practical, that is TS of Cinchona Bark also, isn't it? So always listen these lectures related to the practicals and refer the lab manuals also, so that will be easier for you to understand, isn't it? And listen twice or thrice, isn't it? And you may complete this first experiment also, so that is the first three introductory practicals 1 2 3, and experiment number 4, that is TS of Cinchona Bark, that also you can cover, isn't it? If any doubt difficulty you have, so you can ask, isn't it? Thank you.